So you'll know it's no, you won't know it's recording. So just help me remember. I only see it on my screen. Oh, also, you'll notice as we go along this semester that I love anatomy and physiology. Like, I live and breathe this stuff. And I love to teach it. So sometimes I get a little carried away. I can be a fast talker. So if you need something re-explained, raise your hand and tell me to say it again. Slow me down if you need to, because I will get like worked up, like, ooh, this, ooh, that. And I can tell you about this and that. And so if you need me to slow down so you can write something down, or need me to pause before I move on to the next slide, don't be afraid to stop me. It doesn't hurt my feelings or anything like that. So just let me know what you need, and I'll accommodate as best as I can. All right, so everyone's good with this slide? Everyone got it? Okay. So endocrine system 1, we're going to talk about the important differences between hormonal and neural controls of body functioning. You ended off B32 with the nervous system, which was our first most important control system, but we also have our hormone control system as well. We're going to list the major endocrine organs and describe their locations. It's going to be our lab exercise today. We'll distinguish between hormones and what are called paracrines and autocrines. They're just similar to hormones, but not exactly the same. We'll talk about how they're classified chemically. We have amino acid-based hormones as well as lipid-based hormones, so we'll talk about the difference between those. The two major mechanisms by which hormones bring about their effects on target tissue, and this is determined by the chemicals that they're made up by. We'll identify factors that influence activation of a target cell by a hormone. We'll talk about 3 different kind of interactions of different hormones that are acting on the same cell at the same time. And then we'll explain, or you'll have it actually a PLA at the end on how hormone release is regulated. So that's what we're going over today. So the endocrine system, emphasis on system, is one of the body's 2 major control systems. So everybody remember from B32, your very first lecture, I think it was your first lecture, maybe your second, but the levels of structural organization. Everyone remember that, right? Take a second for me and write down the levels of structural organization in order as you remember them. All right, so what's the first level of structural organization, the simplest? Chemicals is first, yes. Those come to, oh, I hate red. You'll learn I hate writing in red. Red is the worst color. It's so aggressive and I hate it so much. I had a professor back in the day, a biology professor I would write papers for. And I would always go to his office hours for him to read over my paper, and he would say, oh, it looks great. And then I would turn it in, and he would still mark it up with this stupid red pen, and I hated it so much. So I don't, as a rule of thumb, I don't use red. So when you get stuff graded back from me, it'll be in pinks or purples or greens. It will never be red, ever. Before we get started, or actually, after the break, then I'll introduce Shai. But Shai, that's your pal over here. She's amazing. You'll love her. Okay, so chemicals is our first level of structural organization. Chemicals come together to make what? What's next? Organelles. Organelles come before cells. Those chemicals come together to make organelles. Name me an organelle. Mitochondria, powerhouse of the cell. Okay, organelles come together to make cells. Different cell types come together to make tissues. Different tissue types with the same function come together to make organs. Different organs with the same function come together to make, and then the sum of all the organ systems is the organism. So make sure that you have that in the correct order. So if I were to tell you that sickle cell anemia was caused by a mutation in the DNA that caused hemoglobin to be malformed, where does that disease originate from? Which levels of structural organization would sickle cell anemia originate from? Not the cell. Where's the issue? At the chemical level. Hey, if this is a genetic disorder in which there's a mutation in the DNA is not a whole cell. Okay, even though it may be expressed at the cell level with the different shaped hemoglobin and sickled cells, that's not where it originates from. The issue comes from the DNA, and so that disease would originate at the chemical level, not at the cellular level. Does that make sense? Okay. So if you remember this diagram from before, so atoms and molecules, which are your chemicals, come together to make organelles. Organelles make cells, cells make tissues, tissue makes organs, organs make organ systems, and those make the organism. And that's not the first time I'm going to ask you about B32 information today, so just get ready. So the endocrine system, its functions include, well, it's all going to be about regulation and control, but regulation and control of reproduction. So our next lecture, we'll talk about different hormones, and we'll really talk about the reproductive hormones more at the end of the semester. So things like estrogen, progesterone, luteinizing hormone, follicle stimulating hormone. You'll learn about all these different hormones and how they work, mostly at the end of this semester. Endocrine system also controls growth and development. So we'll talk about growth hormone when I see you again on Wednesday. Water balance, we'll talk about antidiuretic hormone on Wednesday as well. Electrolyte and nutrient balance. This is where some B32 review comes in that you should be able to tell me about if you remember. Is calcium homeostasis. What is the hormone you learned about in B32 that helps with calcium homeostasis? That increases calcium when blood calcium is too low. You learned it in the skeletal system. What is it? Yeah, parathyroid hormone. Parathyroid hormone or PTH. I will say for hormones, you can use abbreviations, but that's pretty much the only case in which you can use abbreviations in this course. It's just for the hormones. So parathyroid hormone, or again, you can call it PTH. Now, does anybody remember the targets for PTH? How do we increase calcium levels when it's too low in our blood? Yeah, from bones. So osteoclast is one. We activate osteoclast to break down the bone, which releases the calcium into the blood. What's another target? No, it's an organ. So 3 organs. So one is the bone, osteoclast specifically. What are the 2 other organs that parathyroid hormone targets to increase calcium levels? Are you saying kidneys? I think you're mouthing kidneys back there. So kidneys as well. So when the kidneys are filtering our blood to make urine, they are targeted to reabsorb that calcium back into the blood. So if we need more calcium, just put it back in the blood instead of peeing it out, essentially. And then anybody know the last one? Intestines. So when we're eating food that's rich in calcium and we're low on calcium in the blood, parathyroid hormone will target our intestines so that they absorb even more calcium than they normally would. All right, everyone okay with that? Your parathyroid glands are located behind your thyroid gland, although unfortunately we don't really have a model that shows that very clearly. So it'll most likely be a lecture question rather than a lab question. So we are going to also, with the endocrine system, regulate cellular metabolism and energy balance. So we'll talk about the role of thyroid hormone. And then mobilization of body defenses. We'll talk about this in more detail with the immune system, but there is our thymus gland and a hormone called thymosin that we'll talk about that's going to help with body defenses as well. Questions about the functions of the endocrine system? Okay. All right, so like I said, the nervous system is also a control system of ours. And if my big head is in the way, please let me know that as well. I can scoot over. Again, you will not hurt my feelings. I've been told I have a big head, so I'm used to it. So our endocrine system is made from epithelial tissues. Specifically, we'll talk about the very specific types of epithelial tissue we'll find in glands, which you should already know, but I'll ask you in a little bit. And then the nervous system is obviously made from nervous tissue. Oh, I was going to ask you that. I don't know if you saw it. Anybody remember the very specific type of epithelial tissue you find in glands? Simple cuboidal, yes. And so make sure you remember that. I will probably ask you that again. But simple cuboidal for our glands. How do cells communicate with these two different systems? Is that our endocrine system, how they communicate with each other, those organs, and with the rest of the body, is that hormones are created and sent out by the blood circulates, and those hormones go and find their target organ or target cell. So it's a lot longer process than what we're used to with the nervous system. Those are, now they're not exactly touching, but they're as physically connected as you can be. There's only a very small microscopic synapse in between neurons. And so a much shorter distance for communication with the nervous system. This is local. Ooh, that's not how you spell local. This is local. And this is systemic. One way that the endocrine system works is it has what's called receptor specificity. We do not want hormones to target organs that don't have a role in that particular hormone. And so hormones and their targets have a very specific binding relationship. Do you remember talking about in B32 about enzymes? The enzyme and the substrate fit together perfectly and they have this kind of induced fit. Same thing with hormones and their target cells and those receptors on those target cells. A hormone only binds to a very specific receptor. Otherwise, it will not bind. And we'll talk about that more later. Oh, we'll talk about it more now. So this is on the left is the sending cell or the producing cell here. So this is going to be your endocrine gland cell that is producing the hormone. Now you'll notice that this image says that this is a ligand. Ligand is a general term for a chemical messenger. You learned about ligand-gated cells in B32. It's just a chemical messenger. So that ligand can be a hormone in this case, or a paracrine that we'll talk about, or an autocrine, but it's just a chemical messenger. But that ligand or that hormone is only going to be able to bind to a target cell that has a receptor for it. Once that hormone binds, then a response is carried out. Okay? A non-target cell, a cell that we're not trying to control, will not have a receptor for that particular hormone. And so it won't bind and nothing will happen in that case. Okay? So very specific relationships here. Any questions about that? Okay. Yeah.
Would this work in the same way that blood has like specific receptors depending on the type?
So it's not that the blood cells have receptors. They have these identifying proteins that tell you whether they're type A, type B, things like that.
This would be like a different.
It's very similar. We'll talk about the antibodies and the antigens when it comes to blood typing, how we tell what blood type that person has by the antibodies that get to bind to the antigens is what they're called on the outside. We'll talk about that later. It's a slightly different relationship, but the same concept.
Okay.
Yeah. Okay, so the nervous system, again, is our other control system. Make sure that you do review the breakdown of the nervous system. This is going to come into play throughout the semester, especially the autonomic nervous system. We'll talk a lot about flight or fight versus rest and digest, and how those systems work in different situations and how they affect the different organ systems. So make sure you remember our central nervous system is just the brain and spinal cord. The peripheral nervous system is the nerves that are going to communicate from the central nervous system to the rest of the body. We have a sensory division that brings in information, a motor division that executes out different actions through our effectors. Remember, we have effectors that carry out responses. Anybody remember what the effector is for our somatic nervous system? Somatic means voluntary. So what is the only voluntary tissue that we have? Skeletal muscle, yeah. So that's going to be the effector for the somatic nervous system. And what about the autonomic nervous system? What are all of our involuntary effectors? Specific tissue types. So smooth muscle, yes. What's the other one? Cardiac muscle. And then what's our last effector? You learned at B32. It's going to be glands. Okay, so your autonomic effectors are smooth muscle, cardiac muscle, and glands as well. All right, so since our endocrine system does have to rely on hormones that get sent out in our bloodstream, and those hormones circulate throughout the body until they find their target. This is a slower response than what we get with the nervous system, which relies on electrical impulses that are very fast. It is a slower response time, but it has much longer lasting effects. So when a neurotransmitter is sent out with the nervous system, it's almost immediately terminated. Anybody remember the different types of termination that we have at the synaptic cleft? How do we terminate the effects of a neurotransmitter? Told you guys I was going to ask you a lot today. So I'll remind you, I know it's been a while. How many of you guys, it's been more than a year since you've taken BIOL 32? So how about, was it in the spring last year? You guys all took it? A lot of you. So review the information. It's going to be really important. But how we terminate neurotransmitter activity is one, diffusion. So it'll just diffuse away from the synaptic cleft. Or we have neurotransmitter recycling through reuptake, is what you learned, where we kind of just take it back and we send it out again if necessary. And then also enzymes. So you learned about acetylcholine esterase, breaking down acetylcholine so that we're not contracting longer than we need to. So these effects for the nervous system are very fast. You can send a very fast response, but then it's over very fast as well. Endocrine system, it takes longer to happen, but it lasts much longer as well. Okay, so two different control systems with two different kind of speeds and response times. Where are we at with time? 9:05. We got 20 minutes before we got to take a break. Any questions so far? Okay. And again, let me know if you need me to slow down at all. All right, exocrine versus endocrine glands. This is review. You should know this already. You learned a little bit about glands in B32. So an exocrine gland secretes onto an internal or external surface. You learned all about exocrine glands in B32, like our sweat glands, like the eccrine glands and apocrine glands, or our sebaceous glands, the ceruminous glands with the earwax as well. You learned all about those. Endocrine glands, though, on the other hand, they're going to secrete their substances, hormones, directly into the bloodstream. So exocrine goes on to either an external surface or an internal surface, like your respiratory tract or your digestive tract, while the endocrine glands have direct access to the bloodstream to just drop those into circulation. So which one of these two, and you should know this, which one of these two has ducts to carry their secretions from one place to another? exocrine, right? Because it's supposed to be going to an external or internal surface. So we have to get that product from the gland to whatever that surface is. If it's the salivary glands in your mouth, to get to your tongue or inside your cheeks, or so on and so forth. Endocrine glands, though, have direct access to those capillaries, so no need for ducts. It just dumps it right into the bloodstream. So these ones have ducts, and these ones are ductless. Oh, also you'll notice with my lecture notes, there are like questions throughout. One way that I heard is really helpful is while we're in lecture to kind of just follow along maybe with the PowerPoints and then use the lecture notes as kind of like a review worksheet to go back and be able to answer questions. That's one way to do it. If you're doing it in class while we're going and I pass over a question that you don't know how to answer, or maybe you missed my explanation of it, stop me and say, hey, what's the answer to this question? And I'll let you know. That way you're not trying to figure it out later or struggling with it later. Yeah, so if you need more time to write down those answers or you need me to answer them for you, just let me know. So like I said, our salivary glands will have ducts to take the secretions, the saliva from that particular gland to the oral cavity. You don't need to know the names of these salivary glands yet, but you will when we get to the digestive system a little bit later. Our thyroid gland, which is an endocrine gland, is will be ductless because endocrine glands secrete their hormones directly into the bloodstream. You'll see them surrounded by capillaries. So when a cell from that particular gland makes a hormone, it just dumps it directly into the bloodstream. So no need for ducts with an endocrine gland. Here's a kind of a microscopic at the cellular level. Here are exocrine cells, and they're going to secrete theirs through a duct that's going to go to some kind of surface, versus endocrine, which again have direct access to those capillaries to dump the hormones directly into the bloodstream.
If you get cancer on an endocrine gland, are they more likely to metastasize?
It depends on the location, but we'll talk about like secreting hormones versus non-secreting hormones, because sometimes when you get a tumor on an endocrine gland, it can either allow it to keep its ability to secrete that hormone, then you're going to have imbalances because you'll be secreting too much, or it'll make you lose the ability to secrete and then you're not secreting enough of that hormone. So we'll talk about the differences and kind of the effects that causes later, I think today. All right, so what is a hormone? A hormone is a chemical messenger produced by one set of cells that travel throughout the body via extracellular fluid, like the blood or lymph. To change the metabolic activity of a different set of cells or target cells. So endocrine glands produce the hormone goes out to the target cell and causes some change in that target cell. Okay? Whether it's testosterone causing the growth of an Adam's apple, or growth hormone causing the lengthening of a bone. Okay? These hormones are going to have specific targets to cause specific changes in those organs or cells. If you need a second to write down the definition of the hormone, I'll give you a second before I go on to the next page. Shy, your hair's different.
It is different.
It looks good.
I'm telling you, we're morphing. We change our hair. We said this.
All right. You see how bright my purple guy
Yes,
is?
I love it. You did it half? Or
It's not. I know it's still just a chunk in the front. It'll fade. It'll be blonde again at some point. Anybody still need this definition up? Okay, and I'm sorry if you listen back to the recording and hear me and Shai talking about hair. All right, so before we get into the nitty-gritty of hormones, because we're going to spend a whole 2 lectures, or a lecture and a half now, on hormones, we're going to talk about a very similar chemical messenger that's not quite a hormone, but kind of still gets discussed while we talk about hormones. So we have what are called autocrines and we have paracrines, or you can say we have autocrine signaling and paracrine signaling. What an autocrine is it's a chemical message that gets sent out by a cell and it targets that very same cell. So a cell targets itself. Okay, this happens a lot with the immune system. When we need to make more white blood cells to fight off an infection, those white blood cells will target themselves to make copies of themselves. Okay. Also happens a lot with embryonic development. Cells will target themselves to grow more exponentially to get that growth and development to happen faster. Paracrine signaling is when a cell, here's a signaling cell, sends out a chemical message in its immediate environment, so right around itself, and targets a neighboring cell, so a cell right next to it. Okay, that is paracrine signaling. Why these are not considered hormones is because hormones are classified by the fact that they travel through the blood. This is not going through the blood. Okay? If a cell is targeting itself, it's not going to send it to the blood for it to go all throughout the body and come back to itself. It doesn't make sense. It would be a waste of resources. So it sends it directly out and binds right to itself. It does not need to travel through the blood. Same thing with paracrine signaling. These chemicals are being sent in its immediate environment, just right around itself, and targeting a neighboring cell. Again, does not need to travel through the blood and come back around. It just needs to be in its immediate space in the extracellular fluid or interstitial fluid. So again, they're similar to hormones in which they are sent out, they bind to a receptor just like hormones do, but they do not travel through the blood. Just the cell's immediate space. That's what makes it different from a hormone. Hormones travel long distance. Autocrine and paracrines travel a very short distance, but they work similarly. So with autocrines, the chemical messengers that exert their effects onto the same cell that secreted them. So examples include prostaglandins and smooth muscle. This has to do with reproductive health and the erection. Also the immune system, like I said, T cells or white blood cells, they will target themselves so they can make copies of themselves very quickly during times of infection. I'll show you an image of that as well. And programmed cell death. So we'll talk about a lot of this in the immune system as well. Sometimes when cells are not working to their optimum, when they're not doing a very good job at the functions that they're carrying out, sometimes they'll send a signal to themselves to essentially kill themselves. I'm not doing my job correctly. I'm not providing a usable service to this body. I'm going to send a signal and I'm going to implode, and then the body can recycle my parts. Isn't that really extreme? This happens a lot with the immune system. If a cell is not doing what it's trained to do, it will undergo apoptosis. Well, it tells itself, okay, it's time to implode because we're not doing our job correctly. Okay. It's a sacrifice to the good of the body. Questions about that? Okay. So this is showing you activation of a T cell. Here is what's called an antigen presenting cell, which I'll explain to you a little bit later. It's presenting a foreign piece of protein to a T cell that's resting, not activated yet. So it gets presented with this foreign piece of protein, then it becomes activated, then it sends itself signals. So you see this called IL-2. We'll talk about interleukins later. But it's sending itself this particular signal so then it can make copies of itself. And then it's more prepared to go off and fight off that infection, whatever that foreign thing was that was presented to it. So that is autocrine signaling. It is signaling itself to cause a change. Paracrines, on the other hand, these signals produce locally and are received by neighboring cells, so someone right next to it. They move by diffusion, and these are very short, quick responses. Examples include clot formation that I'll talk to you about in a second, and then synaptic signaling. So when a neuron communicates with another neuron, that's technically paracrine signaling. A signal is being sent out and binding to a neighboring cell. Doesn't have to travel through the blood or anything like that. It's just right next to it, and the neurotransmitter binds to the neighboring cell. So this can be synaptic signaling from nerve to nerve or from nerve to muscle at the neuromuscular junction, as you learned in B32. So I'll give you a second to write notes down if you need to write notes down. I'm going to finish talking about paracrine signaling here in a second. I'm going to give Shai 5 minutes to talk about how we'll take a break, and then we'll come back and finish. Anybody still need this slide up? Okay. So again, neuron to neuron is a form of paracrine signaling. Neurotransmitter gets sent out and binds to either the dendrites or the body of the next neuron in line. That is paracrine signaling. It doesn't travel through the blood. And then clot formation. So paracrine signaling is evident in the very first step of clot formation, which we'll talk about when we get to blood and blood vessels in a few weeks. But the inner lining of our blood vessels is made up of epithelial tissue. The inner lining of any one of our organs is made up of epithelial tissue. And then another layer is smooth muscle in our blood vessels. So when our blood vessels experience an injury, the epithelial cells on the inside send a signal to the smooth muscle right next to it to tell it to contract, to try to close that gap and minimize blood loss for about 20 to 30 minutes while a clot is being formed. So we have an epithelial cell telling the smooth muscle cell right next to it to contract. So that would be also an example of paracrine signaling. If they're right next to each other, it doesn't have to travel through the blood, just to the layer next to it. Does that make sense to everyone? Okay. All right. There's a question on there that says, how are autocrines and paracrines different from hormones? They are different. Ooh, that's not what I meant to do. Here we go. Okay. So how they're different. They are different from hormones because they release chemicals around their immediate space. Not the blood. One thing to remember, though, is that some hormones act as paracrines too. So, for example, our reproductive organs are ones that act, in which hormones can act as paracrines as well. So let's talk about female reproductive organs. So female reproductive organs or female reproductive hormones are produced at the ovaries, right? Your estrogen, your progesterone. At the ovary levels, it helps with cell development and release, which that would be paracrine in that case, because it's all happening just at the ovaries. But then those same hormones are transported to the breast for breast development, transported to the hips for the widening of the hips of the pelvic bones and such. And so in that case, it would be considered a hormone because it's traveling far distance in the body to make things happen, to make a change happen, whether it's mammary glands or whether it's, like I said, hip changes, things like that, fat deposition. That would be considered it acting as a hormone because it's a longer distance. Okay, so some can act as both. All right, so I'm going to go ahead and stop there. I'm going to give Shy, and let me stop the recording, and then I'll resume when we come back. All right, so I turned my recording back on, and I'll put the two videos together before I post it. Next we're going to talk about the different types of hormones that we have depending on their chemical structure. So make sure you see it. Okay. So you all learned in B32 about biomolecules, monomers versus polymers. Anybody remember what the monomer is for a carbohydrate? Monosaccharide. What is the monomer for a protein? Amino acid. What is the monomer for nucleic acid? That one people always take longer to remember that one. Nucleotides, yeah. And then lipids don't really have a monomer, but what do they break down into? It's glycerol and fatty acids is what they break down into. What was the process by which we added monomers together to make a polymer? It had to do with removing a water molecule to do it. Dehydration synthesis. And how do we break down polymers into monomers? Hydrolysis. See, you guys know it. You guys remember. Okay. That's just a review. We're going to focus, though, on proteins and lipids when it comes to hormones. Just want to make sure that you remember all these because it will come back, especially when we get to digestion and things like that. So the structure of a hormone can determine its mode of transport, so how we transport it from one place to another, how long the response lasts, and the types of receptors that it can bind to. So that's all going to be determined by whether it is protein-based, or we call it amino acid-based, or lipid-based. All right, so we're going to do a little bit of a cell review. So again, the structure of a hormone can determine its mode of transport, how long its response lasts, and type of receptors it binds to, because you learn in B32 that structure determines function, right? Okay, so the structure of these hormones are going to determine how it functions. So, not a regular pen. Okay. We're going to talk about protein secretion, which is just more B32 review. Shy is going around really quickly. I'm going to pause for a second. Shy is going to pass y'all a sitting chart. What I want you to do is write down where you're currently sitting. So here, let me explain real quick. Sorry, just forward past this part when you watch the recording. So the bottom is my desk, so it's facing this way. And so this is group 1, group 2, 3, 4, 5, and 6. So just write where you're sitting. And then if you want to stay there, tell me you want to stay where you're sitting, or if you want to move closer or further away, you let me know that. Okay? I'll go ahead and do that. I will try my best to make it work. All right, so protein secretion. How is protein made? Again, B32 review. I want to make sure I'm going in the right order with your lecture notes. All right, so there's something you learned in B32, which, depending on your professor, they may have referred to it as the central dogma of biology or molecular biology, but it's the fact that genetic information goes from this form to that form and then a final form. So it should be that genetic information is passed on from DNA, and then what would be the next step, if anybody knows? RNA, specifically mRNA, and then to what? Protein. What is the process that turns DNA into mRNA? Transcription, yes. And what is the process that changes mRNA into a protein? Translation. Protein. Another bit of review. It's going to be so much review today. What are the specific types of bonds that we find in between the amino acids of a protein? Polypeptide or peptide bonds, yeah. It is a peptide bond that holds them together. Anybody know what kind of a bond that is? A peptide bond? Covalent bond. And so where are proteins made at? Anybody remember which organelle proteins are made at? Ribosomes. Now, we have two types of ribosomes. We have free ribosomes that are free floating in the cytosol or cytoplasm. And then we have attached ribosomes. And attached ribosomes, where are they attached? What are they attached to? The rough ER, and then there are actually some also where? And the nucleus or nuclear envelope. So you'll see some of them here on the nuclear envelope. There's some ribosomes there as well. So as far as the rough ER goes, something else you learned in B32 was the secretory pathway. What is the secretory pathway? We go from the rough ER, what would be the next step? To the Golgi body, to a vesicle, which then leads to exocytosis. Everyone remember that secretory pathway from B32? Okay, so one thing we can say, and as far as the free ribosomes versus attached ribosomes, free ribosomes make protein for cell use. So the free ribosomes are going to make proteins that stay inside that cell, do not leave. The attached ribosomes make proteins for export that follow that secretory pathway. So our attached ribosomes are the ones that are going to be making the amino acid based hormones that we talk about later. Okay, so all hormones are either going to be made from protein or they're going to be made from lipids. And so the ones that are made from protein are made by these attached ribosomes because they will be exported out of the cell, enter the bloodstream to go find its target cell. Now, the other type of hormone that we have is lipid-based hormones. And does anybody remember which organelle does lipid formation or lipid synthesis? The smoothie R. And so smoothie R will be making our other type of hormones, which are our lipid-based hormones. So the functions of the smoothie are lipid synthesis. Anybody remember the other function of the smoothie R?
Breakdown of lipids?
Not well, it could be lipids, but there's a very specific function that smoothie R does. Drug detox or drug breakdown. So our smoothie R, go back. Smoothie R makes the lipid-based hormones. While our attached ribosomes make the amino acid-based hormones. But we'll talk about that more. I'm running out of room over here. Any questions on cell review? I see people still writing, so I'll wait to go to the next slide. Also, if you ever miss anything that I write down, I have these notes that I keep up here. You guys are welcome to come look at them if you miss a page or miss something. I don't gatekeep my notes. If you're here, I don't mind sharing them with you. Okay, anybody still need these notes up? Okay, next slide. So we have amino acid-based hormones, and then we have lipid-based hormones. We're gonna talk about both. So our amino acid-based hormones, they could be as small as a single amino acid. That's what the AA is for. They could be a short peptide sequence, or they can be a full-blown protein with all the levels of structural or levels of protein folding. Amino acid-based hormones are water soluble. Proteins are water soluble in general. And so this means that they travel freely in our fluid compartment, so they can travel through blood and other fluid without any problems. Would a amino acid-based hormone, though, be able to cross a cell's plasma membrane if it's water soluble?
Yes.
No, it will not. Okay. Why? What is within the plasma membrane that would prevent something that's water soluble from crossing? The phospholipid bilayer. The phospholipid bilayer has these hydrophobic tails, if you remember. I'll draw this in a different color. Not the prettiest phospholipid bilayer, but it's a bilayer. So this portion here on the inside is hydrophobic. The phosphate heads are hydrophilic. So let's say this little blue dot here was an amino acid-based hormone. If it tried to cross a plasma membrane, once it gets to the hydrophobic portion in the middle, which is the bulk of what that middle of the plasma membrane is, it's just going to bounce it right back out. Okay? The hydrophilic amino acid and the hydrophobic tails are not going to be able to interact. Yeah?
Can you use channel protein?
Oh, yeah, of course. Yes, but can they cross on their own? No, they cannot. Okay? Now, because they can't cross the plasma membrane, what they're going to do is they have actually receptors on the outside of the cell, so they don't even have to cross. They're going to have a receptor on the outside of their target cell. They're going to be able to bind from the outside and allow for changes to happen. These are generally faster acting than our lipid-based hormones, and we'll talk about why. They just have a very complex process because of their structure, and we'll talk about that later. But these are generally faster acting. Amino acid-based hormones can act over minutes to hours, while lipid-based, which we'll talk about later, is more days to weeks. Okay, these are much faster acting. And again, they're faster acting because they bind to a receptor on the cell surface. There's no need to cross the plasma membrane. They just bind straight from the outside. There is one exception, though, and that is thyroid hormone. So I said before, we kind of have different sizes of amino acid-based hormones. It can be a large protein hormone like insulin that is made up of about 51 different amino acids. It could be oxytocin, which is a pretty short string of amino acids. Or thyroid hormone is actually derived from a single amino acid. Very, very small, just one amino acid. Okay. We do have two different thyroid hormones, T3 and T4. I don't need you to know the difference. The one that gets thrown around most often is thyroxine, which is T4. But again, I don't need you to know the difference. So if you tell me thyroxine or thyroid hormone. It really doesn't matter to me. You should know what the T3 and T4 mean. One of the requirements to make our thyroid hormone is iodine. It's part of the reason why our salt is iodized, because we need that iodine to make thyroid hormone to keep our metabolism up. And so our T3 thyroid hormone is made from 3 iodines. Our T4 thyroid hormone is made from 4 iodines. So a clinical application to that is something called a goiter. Okay. So this is a female from another country that has a goiter. Possibly the salt that she's using or seasonings they're using is not iodized. For some reason, she's not getting enough iodine in her diet, which can cause a goiter. So a goiter is an enlargement of the thyroid gland. Essentially, her thyroid gland is overworking to try to produce thyroid hormone, but it doesn't have enough of the materials to do so. Okay? And so what she's exhibiting here with the enlarged thyroid is a goiter. Now, over time, this can lead to hypothyroidism, because if she doesn't have enough of the materials to make enough thyroid hormone, then she's not telling her cells enough to metabolize, or to increase her metabolism in times of need. So most likely she's also experiencing some hypothyroidism, but the enlargement of the thyroid gland is called a goiter. Okay? And again, this is due to iodine deficiency.
So does iodine just help like release the hormone?
No, it's one of the requirements. So in order to make the thyroid hormone, you have to have the iodine, or else it's not.
Okay.
You change the structure, you change the function. It's not going to work the way that it should without the
What
iodine.
causes the enlargement of the thyroid due to the?
I think it's just an overworking of those cells. They're trying to produce as much thyroid hormone as they can, but they don't have enough materials, and it causes an inflammation and enlargement of the gland.
Does it send the signal to the body to absorb more iodine?
It doesn't really work, because iodine is something that we need in very small levels, and so we wouldn't really be absorbing more. We just need to have a lot at one time, or a certain amount, but it's very little. But if we don't have enough, then it causes these issues. Yeah.
So is goiter a result of like hypothyroidism?
A goiter would lead to hypothyroidism. Because having an extended period of time without being able to make appropriate amounts of thyroid hormone, then you're going to see slowing down in your metabolism. One thing I forgot to mention, or I forgot to elaborate on. So our amino acid-based hormones, like I said, they generally act from the outside of the cell, because amino acid-based things can't cross the plasma membrane because they're water soluble, and the hydrophobic tails wouldn't allow them to do so. But our thyroid hormone, which is only a single amino acid, large, so very, very small, those ones actually do cross the plasma membrane. Okay, because they're very small, they can kind of swim their way through the hydrophobic tails without being seen, okay, without causing a bouncing back effect. So they're the only ones. Thyroid hormone is the only exception and the only hormone that can cross, that's amino acid based, that can cross the plasma membrane. Okay, that's the one exception. Because it wouldn't be anatomy if there wasn't exceptions to everything and little details for you to remember. Okay, any questions on amino acid-based hormones? Okay. Lipid-based hormones, on the other hand, okay, you're going to see a lot of ring-type structures because they're lipid-based. If you hear steroid-based hormone, same thing. Okay, lipid-based hormone, steroid-based hormone are interchangeable. I think your book may use steroid-based, maybe. But either way, they both mean the same thing. So you can use lipid-based or you could use steroid-based. Because they really are steroid-based, but steroids are a type of lipid, so they both work. So these are not water-soluble. So when they are traveling through the blood, they need to have a protein chaperone to take them from place to place, because lipid-based things do not like to interact with water. And our body is made up of mostly water. The fluid in between our cells, our bloodstream, all of that is water-based. And so anytime a lipid-based chemical is traveling from one place to another, it's usually bound to a protein that can chaperone it through those fluid spaces that it doesn't particularly like. The lipid-based hormone, the receptor is inside of the target cell. Why? Because lipid-based things can cross the plasma membrane, and they will. Okay, these lipid-based hormones will be able to cross through the phospholipid bilayer to be able to go into the cell. And so how do target cells get receptors? We'll talk about that next. Before we get there, any questions on lipid-based hormones? So they are not water soluble. They are hydrophobic, so they can go across that hydrophobic plasma membrane in order to get to the inside of the cell. We'll talk about what they do when they get there. So we'll talk about next again, how do target cells get those receptors? So receptors are made from protein. Right? All these receptors on the outside of the cells are made from protein. Where did the directions to make a protein ultimately come from? DNA, right? So how do cells get the receptors? They get it from a gene, essentially our DNA. This gene is going to be read by an attached ribosome if receptor is on the surface. And this gene will be read by a free ribosome if it's an internal receptor. Okay, so we talked about how we have free ribosomes and attached ribosomes. Attached ribosomes are going to be making proteins for export and really also for embedding into the membrane as well. So if we have a receptor, a hormone receptor that's on the surface, that receptor is being made by an attached ribosome because it has to go to the outside of the cell. Okay? And amino acid-based hormones would be attaching to these outside receptors. If the receptor is internal, inside the cell, then it's going to be made by a free ribosome because free ribosomes make proteins for inside the cell. Yeah?
What if it's like in the middle, like half outside, half inside?
What do you mean?
Like channel protein?
Oh, yeah. So the receptor itself will always be on the outside. Whether it's attached to another protein or not, it's always going to be on the outside for a surface receptor.
And that's made by
That'd be attached ribosome, yeah. Now, our lipid-based hormones, like we said, they can cross the plasma membrane, so they go inside for the receptor. So whenever we have an internal receptor, that's made by a free ribosome because those make proteins to stay inside the cell. And we'll talk about those different mechanisms next. Anyway, still need to write this stuff down. So lipid-based hormones, when they do bind to a receptor, and this is going to be on the inside of their target cell because they have the ability to cross that plasma membrane, their process is going to be a little bit more complicated. So how lipid-based hormones work is they go into their target cell, okay? They bind to a region of the DNA to turn on a gene to make another protein that's going to carry out that response, whether it's an enzyme or some other chemical. And so again, when a hormone binds to the receptor on the inside of the cell, the complex activates genes, and I'll show you how that works. So they're generally slower acting. These can work, so it takes a while for it to happen because it has to go into the cell, find that very particular piece of DNA to turn on that gene to make the protein, but it's harder to shut off. So these can work over days to weeks. So longer effects. So it takes longer to happen, but the effects are longer. Amino acid-based hormones that bind from the outside, it's a very, very quick response and only lasts for minutes to hours. Yeah.
Like puberty
Yeah. So like the hormones that regulate our cycles. So female cycles, ovarian cycle, or menstrual cycle, or like you said, puberty. Those things take long amounts of times. Could be days to weeks for these things. So those would be lipid based. And all of our reproductive hormones are lipid based. Your estrogen, your progesterone, your testosterone, all of that is lipid based. They generally work longer than other hormones do. So something like insulin that just lowers your blood sugar, right? And so things like that are going to be happening very fast, minutes to hours. So those would be more amino acid based. All right, so hormones are either going to act through what's called a second messenger system, which are amino acid based hormones are going to do, the ones that work from the outside. Or they're going to activate genes, which is what our lipid-based hormones do because they go into the cell, find a piece of DNA, activate a gene, and then create a protein that's going to carry out the response. So we'll talk about how those things work. So again, for our amino acid-based hormones, they can't cross the plasma membrane, so they're going to bind from the outside, and that's going to cause a response on the inside. If it's lipid-based, they're actually going to cross, find a receptor on the inside, and cause a gene activation. And again, that's going to take a little bit longer. And then any non-target cell is not going to have a receptor for that particular hormone. Okay? Hormone target cells have a very specific relationship. All right. So a target cell's response is going to depend on the tissue type. Okay? Muscle cells can only contract, glands can only secrete, so it's all going to depend on what that tissue type or cell type is. So muscle cells, if they are the target, they are just going to increase or decrease the amount of contraction that's happening. If it's an epithelial cell that's being targeted, it's going to increase or decrease the amount of secretion that's happening. It's all going to depend on what that particular cell or organ is. Common cell responses include opening and closing ion channels, which may increase or decrease plasma membrane potential. With that example, all it's going to do is we're either going to have threshold be met more quickly, so the response can happen faster, or more slowly, so it makes it harder for that response to happen. Okay. It can help stimulate the activity or synthesis of enzymes, which will then carry out whatever the desired function is or response. Or like in the case of growth hormone, it can stimulate mitosis, leading to repair, replacement, and growth of tissues. It's all going to depend on what the target cell is and what their normal function is. So this is how amino acid-based hormones work. Okay, I'm going to show you the long version, and then I'm going to tell you what you're responsible for. Okay, so it's a recording. I'll draw on this thing. Okay, so again, this is for amino acid-based hormones. These ones do not cross the plasma membrane, so they're going to be working from the outside. Okay, so you can see the hormone is the first messenger. This whole thing is called the second messenger system. So our hormone is our first messenger. It's going to bind to a receptor on the outside of the cell. Okay, that's where it's binding. That is going to activate something called a G protein. That G protein, once it's activated, because remember, if you change the structure, you change kind of the function, what's going to happen. So when you're not going to be able to know what I'm talking about if I'm using the screen up here in front. But as soon as that hormone binds there, like we said with the enzyme and the substrate, it has kind of an induced fit. So it wraps itself around that hormone, which is going to change the overall shape of that particular protein. And so that's what causes it to be able to activate this G protein here. And then that G protein is going to move across the plasma membrane, bind to an enzyme here. It's called adenylate cyclase. You don't have to know that, so don't freak out about enzyme names. I'll let you know when you have to memorize something. But it's going to activate an enzyme, which is going to cause the production of this second messenger. This is our second messenger. Most of the time, CAMP, which is called cyclic AMP, adenosine monophosphate. You guys all know about ATP, adenosine triphosphate. That's this guy here. We use that, take off a couple of phosphates, and now we have what's called cyclic adenosine monophosphate. So we just take off a couple of phosphates and now we have this second messenger, which is then going to carry out whatever the desired response is. Okay. So again, the hormone binds to the receptor, causes a conformation change, which activates the G protein. The G protein slides across the plasma membrane, activates the enzyme, and then the enzyme is going to create that second messenger, that cyclic AMP. And then the cyclic AMP is going to trigger responses of that target cell, which could be making enzyme, which could be stimulating secretion, which could open or close ion channels, or any one of those examples that we've given you. Now, what you're responsible for is just this summary here. How amino acid-based hormones work is we have the hormone, passes it off to the receptor, which passes it off to the G protein, and then to the enzyme, and then finally to the second messenger, which is then going to carry out the response. Okay, that's kind of the flow of how amino acid-based hormones work. The reason why these hormones are a lot faster acting than the lipid-based hormones is because all these materials here are already produced. They're already ready to go. So as soon as that hormone binds, it all happens automatically. And it's going to be very quick. So this is why these types of hormones act over minutes to hours. Okay, very quick responses that are turned off very quickly as well. Any questions about how that works? Okay. So the advantage is it's a quick on and off situation here. That's an advantage. We can quickly cause this change to happen. The internal ingredients are already made and it's ready to go as soon as that hormone lands. So very quick response. The disadvantage, though, is that if we wanted a longer term response for this particular hormone, we'd have to keep producing that hormone and keep it binding in order to keep that response going, which is going to cost materials, going to cost energy. Okay? So it's a slight disadvantage to the system. One other thing to note, yes, CAMP is the second messenger we use very often, but it's not the only second messenger we use. Calcium is often also used as a second messenger, especially when it comes to oxytocin, which we'll talk about a little bit later. But because it's used as a second messenger, we generally keep blood calcium levels pretty low, because if we had an excess of calcium, we may be causing unwanted reactions throughout the body, and we don't want that to happen. So we generally keep blood levels of calcium low because it can act as a second messenger in these systems. And there's actually some amino acid-based hormones like insulin that don't even use second messengers at all. Okay, so there's always going to be exceptions to these general rules. Any questions about how amino acid-based hormones work? Okay. All right, so lipid-based hormones. So this is going to be the case for lipid-based hormones as well as that thyroid hormone that I talked to you about. Because it's made from a single amino acid, very small, it's going to be able to cross the plasma membrane, unlike the other types. Okay? So lipid-based hormones plus thyroxine, or you can call it thyroid hormone, which is an exception to our rule. They are going to use this method. So because these lipid hormones and thyroid hormone can cross the plasma membrane, here is that steroid or lipid-based hormone. Again, we can call it lipid-based. And then also our thyroid hormone will act in this way as well. But that hormone is going to cross the plasma membrane. And then here is a receptor protein on the inside of the cell. So because it's internal, it was made by a free ribosome, not an attached ribosome. But you can see the receptor on the inside of the cell. The hormone is going to find that receptor and bind to it and create a complex. That complex is going to be able to travel into the nucleus in order to find our target gene or target piece of DNA. And it's going to activate that gene. Once the gene is activated, we can transcribe it into mRNA, translate it into a new protein, and then that new protein can carry out the desired response. Okay. So again, steroid hormone diffuses through the plasma membrane and binds to an intracellular receptor. It becomes a complex, which then enters the nucleus. The complex then binds to a very specific DNA sequence, we have transcription, translation, and again, that new protein is going to carry out the response. It could be an enzyme or some other chemical doing the work of that cell. Any questions about how lipid-based hormones work? You're going to draw out this diagram and show me the steps so you'll have some practice with the steps of this for today's lab, which will be mostly homework because we're already at 10:15. So when we do make these proteins, that's happening after we activate the gene, transcription, then translation. Don't forget, translation is a whole process that you learn in B32. Anybody remember the specific kind of RNA that brings the amino acids to the growing chain? tRNA, right? Don't worry, I'm not going to test you on translation. You were already tested on it last semester, but just make sure you review and you understand how proteins are made. Once the protein is made, it's folded. Remember, we have primary, secondary, tertiary, quaternary levels of organization for protein. So it gets folded into its protein shape. It's put into a vesicle to be given to the Golgi body. The Golgi body does final preparations, puts it in its own vesicle to be transported to the plasma membrane, and then sent out by exocytosis. All review. And then remember, these lipid-based hormones are slower acting. They work over days to weeks versus amino acid-based that work over minutes to hours. And again, this is mostly our reproductive hormones that are lipid-based. We'll talk about some other exceptions too. So why are hormones released? They are to maintain homeostasis, to keep everything regulated, whether it's blood sugar levels or blood calcium levels or speed of development. Lots of different things that are being controlled by hormones. So there are 3 types of stimuli that cause hormone release, and this is going to be your PLA for today. And actually, I'll go to the end of the slide so I can just show you now so I don't forget. Let's see. So your post-lecture assignment for today, and we'll go back and we'll talk more lecture, but your post-lecture assignment for today is to learn about how we stimulate an endocrine gland to release its hormones. So for example, humoral stimulus. So we just talked about parathyroid hormone. When your blood calcium levels are low, we release parathyroid hormone to target your bones to release some calcium, to target your kidneys to reabsorb some calcium, target your intestines to also absorb some calcium. So that would be considered a humoral stimulus, where there's a drop in something in the blood, which triggers the release of the hormone. Okay, same thing for something like insulin. When your sugar levels are too high, your body detects that as a change in the blood, and then we release insulin. Okay? Or on the opposite end, when blood glucose levels are too low, your body detects that change in the blood and then releases a different hormone called glucagon. That would be a humoral stimulus because it's a change in something in the blood that causes the release of the hormone. Okay? A neural stimulus is when the stimulus or change comes from the nervous system. So this is an example of like your fight or flight response. You get scared or you get embarrassed or what have you. There's some emergency situation that causes a signal in your brain to then cause the release of epinephrine and norepinephrine. That is a neural stimulus. Or when you start having baby move down towards the birth canal and you have stretching in your smooth muscle of the uterus, that causes signals to be sent, oxytocin to be sent out and make your uterus contract even more. That is a neural stimulus. Okay? The contracting of your muscles sends a signal to your brain to then release more oxytocin. Or oxytocin is also used for breast milk letdown. So when baby suckles on the mom's teat, that's going to cause oxytocin to get released because of the stimulus of those receptors, and then milk can be let down. That is a neural stimulus. Hormonal stimulus is when a hormone tells another hormone to be released. That's going to be what we talk about a lot on Thursday. We have something called growth hormone releasing hormone. So that gets released before growth hormone can get released. Thyroid releasing hormone is released before thyroid stimulating can be released, and then thyroid hormone can be released. We'll talk about all those pathways. But that's when a hormone controls the release of the next hormone. It's like a hormone sequence. Okay. So those are the three ways that we cause endocrine glands to release their hormones. Either it's a change in the blood, which is called a humoral stimulus. It is a signal from your nervous system, like you said with the suckling or muscle contraction. That's a neural stimulus or your fight or flight response. Or it's a hormone sequence where one hormone tells another hormone to be released. Okay, so you'll learn about that in your textbook. Give me some definitions and some examples at the end of today's lecture in the lecture notes. And you'll turn that in when I see you again on Wednesday. I need to get my Monday, Wednesday, Tuesday, Thursdays all correct. I'm going to see you on Wednesday. So cells respond to a hormone if they have a receptor for that hormone. These are called target cells. So what receptors a cell has determines whether a hormone will bind to and activate it. So some receptors are expressed on the membrane of most body cells. So thyroxine, thyroid hormone, that regulates your metabolism. All your cells have the ability to metabolize. So all of your cells will have receptors for thyroxine or thyroid hormone. Okay. On the other hand, there are some hormones that are very specific and only target a very specific area of the body. We have another hormone that we'll talk very little about, but we'll still mention it. But this is just to show you how specific it can be. We have something called adrenocorticotropic hormone. It regulates the release of our cortisol, which is our stress hormone, but it only targets the cortex or the outside of the adrenal gland. So super specific. So we're only going to have very few cells that have a receptor for adrenocorticotropic hormone. It's on a very specific region of a very specific organ versus thyroxine, which will have receptors all throughout the body. And so you'll have to learn, and we'll talk about it when we get back on Wednesday. You'll have to know what the hormone is, what it targets, and what type of cells it's going to be binding to. So we'll get through to that when we get to Wednesday. Target cell interaction with the hormone depends on three things. So a target... Bye, guys. She's leaving. She'll be more helpful on Wednesday. I took up a lot of time in the beginning. So how well a cell responds is going to depend on a few different factors. One is circulating blood levels. The more hormone you have around, the larger your response you can give, to a point though. There comes a point though when more hormone doesn't necessarily mean a bigger response, because there's only so many receptors for those hormones. So once we're saturated and used up all the receptors, it doesn't matter how much more hormone you release. It's going to have a certain stopping point where you can't increase the rate of response anymore. Does that make sense? Okay. Also, the number of receptors on or in a target cell available for binding. So our bodies, which is really interesting and really cool, our body has the ability to increase the number of receptors when we want a big response and decrease the number of receptors when we want a smaller response. That is called downregulation and upregulation. So I'll explain that to you. So response by a cell to decrease the number of receptors for a certain hormone leads to a decreased response as less binding sites are available. That is called downregulation. So GnRH, which we'll talk more about, you don't really need to know the ins and outs of that hormone just yet. GnRH stands for gonadotropin releasing hormone. It's going to cause the release of our sex hormones, essentially. Okay? But we only need a certain level of sex hormones. We only need a certain level of testosterone, estrogen, progesterone, etc. There comes a point where it's too much hormone and we don't want that, right? So here's GnRH. It binds to the cell and then causes the release of other sex hormones. If our sex hormones are at a level that's normal. We don't want to exceed that. If we're getting to the high end of our normal range, what's going to happen is the target cells are going to decrease the number of receptors, so it binds less often, so you're making less hormone until those levels kind of come down and normalize. Okay? And then when levels get too low, then we'll increase the number of receptors, so then it binds more often, we make more hormone. One example of that, so upregulation is the response by a cell to produce more receptors for certain hormones, so its response increases due to more binding sites for the hormone. So one example is FSH. So with this one here we have, so follicle stimulating hormone is one of our sex hormones. LH, luteinizing hormone, is another one as well. So one thing is, in order for ovulation to occur in a female cycle, and we'll talk about this at the very end of the semester, we have to have something called an LH surge. There has to be a really big increase in the response of LH in order to cause ovulation to occur. So at that time, the number of receptors is going to increase to allow for that surge and to allow ovulation to happen. So there are times in our journey as humans where we're going to need more hormone or less hormone, and we can upregulate or downregulate for that. Okay? There are certain cancers that kind of feed off of estrogen. Estrogen can cause more growth, which can cause the increase of cancer-related growth. So we can prescribe downregulators. So estrogen binds left often, you get less of an effect. It can slow the growth of some tumors. And so we can actually prescribe downregulators as well. So our body does it ourselves, and we can also prescribe those things to patients as well. Does this make sense? Also affinity. So the strength of binding between a hormone and the receptor is going to determine how strong that bond is. So some chemicals are more attracted to each other than others, or some bind more tightly to each other than others do. Okay? And so some artificial hormone compounds, so if you're taking hormone replacement therapy, sometimes those artificial hormones don't bind as tightly as the natural occurring ones do, and so you may get less of a response with hormone medication management than you would if your body was making it normally. Okay? So there's a connection or a relationship between the hormone and the receptor. And some bind more tightly than others, and so therefore they'll have a bigger response than others. Questions about that? Half-life. Oh my God, there's so much to cover. I'm so sorry, y'all. Half-life. So hormones circulating in the blood are either going to be amino acid based or lipid based. So amino acid based are the ones that are water soluble. They bind from the outside. They work very quickly, over minutes to hours. Lipid based, because they're not water soluble, they can cross the plasma membrane. They're lipid soluble, so they can cross. They bind on the inside, which makes them activate genes, which is just a much longer process. And so these work over days to weeks. Now, because amino acid based hormones work very quickly, and they're amino acid based and they are water soluble, they're actually removed really quickly by the kidney. So we urinate those hormones out really quickly after their use. Lipid based, though, because they are lipid based, they're actually processed by the liver. The liver breaks down lipids, which is just a much longer process than the kidneys just excreting them. Okay? For a lipid base, they have to be broken down and recycled by the liver, which may take weeks to happen. So that's why it takes longer for these to shut off, because it takes longer for us to break those things down and get rid of them. So a half-life, I keep forgetting which one I'm working on. A half-life is the length of time for a hormone's blood level to decrease by half. So if we have 80 milligrams per deciliter circulating, how long does it take to get it to 40 milligrams per deciliter? Now, if you had, so let's say we're talking about amino acid-based hormones. So we're talking about the kidneys, getting rid of them. What if your kidney was diseased? Would that cause an increase or decrease in the half-life of that particular hormone if your kidney is diseased and not working as well? It would increase the half-life. It would take longer for us to get rid of that particular hormone. Okay. Same thing with our liver. If our liver was diseased, for the lipid-based hormones, it would increase that half-life because it would take longer for us to process those and get rid of those. Does that make sense? That will be a question that comes up in your near future, so make sure you have that wrapped around your head correctly. All right. Really quickly, urine versus blood tests. Urine samples can tell us yes, something is present, or no, it's not. It's not going to tell us very specifics about the levels that you're going to find in your body. If we want to find out particular levels of circulating hormones, we would rely on a blood test. Okay, so urine test will tell us yes or no, is it present? Blood test will tell us more about the concentrations of those particular hormones. All right. Types of interactions of 2 or more hormones in the same target cell. So this is when we have, again, 2 hormones working on one cell. Let me find my notes for that real quick. Okay. So we'll talk about antagonism first. So antagonism is when 2 hormones are working on the same target cell, but they have opposite effects. So an example of antagonism is insulin versus glucagon, which we'll talk about more on Wednesday. Glucagon. So insulin is what gets released when our blood sugar is too high. So what insulin targets our body cells and tells them, hey, there's extra sugar, take that extra sugar to make more energy if you need it. Or it will target our liver to take that sugar out of the blood and store it in the liver as glycogen, which we'll talk about later. Glucagon does the very opposite. Glucagon is going to tell the liver to break down that glycogen and release more glucose in the blood. Okay? So they both work on the same target cells, the liver in this case, but they're going to have different effects, opposite effects. Okay? Most of the time you're going to have both hormones circulating at any one time, but which one is the one that's dominating? Which is the one that's doing more work? Okay? And so these would have an antagonistic relationship with each other because they're doing the opposite things. Ooh, do not fall. Okay, synergism. Synergism is when the combined effects, so these are 2 hormones working on the same target cell still, but when the combined effects are amplified. One example is glucagon and epinephrine. Both cause the liver to release glucose. But together, they release 150% of the glucose that either can alone. So the amount of glucose that's released when they're both working together is much greater than any one of them working by themselves. Yeah.
Would hormonal stimulus fall into this?
No, hormonal stimulus is how do we get a gland to release a hormone. This is how do 2 hormones interact when they're working on the same cell. Okay. So one thing to remember, because a lot of people get these things mixed up. So on an exam, if I ask you to give me the hormone interaction, if I'm asking for an interaction, it can only be permissiveness or synergism or antagonism. If I'm asking you for a type of stimulus, it can only be the humoral, hormonal, or neural that we talked about before. So try to keep your mind focused on these key terms to help you kind of use process of elimination to figure out, okay, if I'm asking for an interaction, it can only be these things. I'm eliminating everything else. It can only be one of these three terms. If I'm asking for a stimulus, it can only be one of those other three terms. So make sure, that's where like flashcards might be good, or like Quizlets, interactions, 1, 2, 3, stimuli, 1, 2, 3, so that you kind of don't get those two mixed up, because a lot of people do get those two concepts mixed up. Everyone okay with antagonism and synergism? Okay. Last one is permissiveness. Oops. Permissiveness is when one hormone is dependent on the other. So examples for this. Okay. Is that thyroid hormone is needed for reproductive development in embryos and fetuses. So basically, our reproductive hormones, like our progesterone, estrogen, testosterone, those things, or actually their precursors are called androgens, which we'll talk about later. In order for these androgens to work, there has to be enough thyroid hormone circulating. Okay, so the effects of these reproductive hormones depends on the presence of thyroid hormone, which is why thyroid hormone is one of the levels that are highly looked at when you're doing prenatal care. You need to make sure you have enough thyroid hormone in order to make sure that your baby's properly developing. All right, questions about that? Here's a critical thinking question here. So it says that the female hormone estrogen is known to increase the growth rate of certain types of tumors involved in breast cancer, while other types of breast cancers do not respond to hormones at all. Think of some treatment options for these two different types of breast cancer. I'm dumb and I already talked about it when I should have had you guys thinking about it. I already talked about downregulators, so I'll kind of explain this a little exercise with you guys. So again, we have some cancerous tumors that respond to hormones, so more estrogen means more growth, some that don't respond at all. Okay, so those that respond to estrogen, right, and it causes growth, we may want to prescribe them downregulators. Okay, ones that don't respond to hormones at all, we may want to try something like chemotherapy or surgery or other options. But if it's a hormone reaction, we may want to just prescribe them downregulators, slow down the growth of that tumor, possibly excise and avoid the need for chemotherapy or radiation. Some people may say, well, if it's a hormone issue and it's responding to levels of hormone, well, why don't you just do a hysterectomy and take everything out so that you're not producing the hormone anymore? That is an option. Although people are experiencing breast cancer at younger and younger ages now, that you want to take in that patient's kind of wants, desires for the future into consideration as well. Do they want to have children? Because you may not want to go that drastic on a patient that's very young. You don't want to remove their ovaries if they still desire to have children. So you want to think of different options, which in that case, maybe we do want to give them a downregulator so the estrogen isn't causing such a increased growth of that particular tumor. Okay. Does that make sense? Okay. All right, so, that's it, we're done. Okay, so this is your post-lecture assignment, is the 3 different types of stimuli. You're going to give me a definition and examples of each. I know today was a lot. Endocrine 1 is just a long lecture, and then we spent a lot of time on the syllabus.